Assalamu alaikum, everybody. I hope you're all doing well, inshallah. My name is Muhsin Ali. I am a second year medical student, and today, inshallah, I will be giving you guys the PAL on enzymology. This is going to cover the three lectures you took with Dr. Ahmed Al Jada and Dr. Raja, the enzymes 1, which is Introduction to Enzymes, enzymes 2, which is enzyme kinetics, and then lastly, clinical enzymology. So before I get started, I'd just like to say that this thing is like these lectures are very quite simple, I would say. There's just a few things you need to understand here and there. Of course, do a lot of questions after you finish the lecture, and then inshallah you should be good for your midterm or final. All right. So of course, if you have any questions at all, feel free to write in the chat. Let's begin. All right, Bismillah. So before we get started with the actual lectures, there's just a few definitions that you need to be familiar with. So hold up just a second, guys. Okay. So there's just a few definitions. So first of all, enzymes, and this is like very basic stuff. Enzymes are biological catalysts of specific biochemical reactions, and a catalyst is just simply a molecule or an agent that is going to speed up the reaction without being changed itself. So it's going to speed up the reaction of that specific reaction that it's catalyzing, but it's not going to change chemically. It's going to stay the same. Now, the active site, as you guys should already know from your normal high school chemistry, is part of an enzyme, a specific part of the enzyme where the substrate is going to bind, and the substrate is simply just the reactants. So you can imagine it, for example, like this. You have your enzyme. This is your active site, a part of the enzyme. You have your substrate, which is the reactant, and it's going to bind in order to perform its function. Now, specificity. Before I read this, we will get into the details of specificity and all of these things later on. So don't mind that. So specificity just refers to how specific an enzyme is to a specific reaction. So an example of this is lactase breaking down lactose. So lactase is only going to break down lactose because the substrate is specific to the active site of the enzyme. Okay. Now, what Dr. Ahmed mentioned, the way that you can remember or the way you can know that an enzyme is in fact an enzyme is because it ends with this ase over here. So lactase, you know that that's the enzyme. And lastly, a zymogen is simply just an inactive precursor of the enzyme. So you have the precursor, which is inactive, will get converted into the active form of the enzyme. And this is what you call the zymogen, which is the inactive part. And again, an example of this would be pepsinogen, which is going to be converted into pepsin. Now, again, the way you can know whether or not this is a zymogen is because it ends with this ogen over here. So pepsinogen into pepsin, trypsinogen, trypsin, same idea. All right. So now this is the graph that shows you exactly what the difference is between a catalyzed reaction and a uncatalyzed reaction, or a reaction that has an enzyme and that doesn't have an enzyme. So before we do that, let's just quickly orient ourselves. You have the free energy here on your y-axis, and you have the progress of the reaction on your x-axis. So what you can see is that with the enzyme, which is the purple one over here, you see that the free energy of activation over here is a lot lower than the free energy of activation with the uncatalyzed. Can you see my cursor, guys, by the way? Let me just use a pointer. Hold up. Okay, I hope you can see my cursor. I don't know what's happening. Okay, great. So you see that the free energy of activation for your catalyzed reaction with the enzyme in purple, which is from the reactants all the way to the peak of the reaction itself, is a lot lower. Now, what your free energy of activation is and why it is a lot lower is because it's the free energy of activation is the energy that the reaction needs in order to go from the reactants. So over here, that's why it starts at this line where the reactants are, to the top, to the peak of the reaction, which is what we call your transition state, right? So that is what your free energy of activation is. It's the energy needed to go from the reactants up to the transition state, and it is a lot lower in a catalyzed reaction compared to a non-catalyzed. Now you also see over here this green arrow, which shows the free energy of the overall reaction. So now we're not looking at the energy of activation, but we're looking at the overall energy. And again, you can see this because it goes from the reactants this time all the way to the product to the products, not to the transition state, which we saw over here with the free energy of activation. And what you need to remember is that the free energy of the overall reaction in a catalyzed or uncatalyzed does not change. So the free overall energy of the reaction is the same. Okay, just to put that into words, you have your enzyme is going to decrease the free energy of activation, which is the energy needed to reach T state, which is the transition. So we said you go from the reactants, then you go into a transition state, and then to the products. And the energy to go from the reactants to the transition is what we call your free energy of activation, which is decreased in catalyzed reactions. And then, of course, you need to know and remember as well that there is no change in the overall free energy of the reaction, which is signified by this weird symbols over here. All right. So of course, in order for the enzyme to elicit its function, there needs to be the binding of the substrate to the enzyme's active site. And again, we mentioned the active site is in the enzyme, and the substrate is the reactant. So there's two hypotheses that you can kind of say that show this enzyme substrate complex or how it happens. You have the first one, which is the lock and key hypothesis, and then you have your second one, which is the induced fit. Like the name suggests, the lock and key is like a lock and key, right? Who would have guessed? You have the lock, which is specific to the key. So this means that there's an exact fit of the substrate to the active site. So again, let's orient ourselves real quick. This darker shade of blue is your enzyme with the active site, and then this lighter shade is your substrate. And you see that there is an exact the fit of the substrate to the the fit of the substrate to the active site of the enzyme is exact. So then you have the binding, and that's the exact fit for the lock and key hypothesis. Now, on the other hand, Dr. Ahmed mentioned that this is the more realistic kind of hypothesis, is the induced fit. And this is where there's a conformational change that happens in the active site. So you can see now the substrate and the active site are not exact fit. You see that there's this little bulging out on the substrate this time. But what happens is when it binds, there's going to be a conformational change in the active site, which is just the proteins and the amino acids, they rearrange themselves in order to fit the substrate. So again, lock and key hypothesis means that there is an exact fit between the substrate and the active site of the enzyme. And an induced fit, it's that there is a conformational change. So it's not exact. It changes once the substrate is able to bind to the active site. All right. So now we go back to the specificity that we talked about. What did we say before? We said that it is how a specific enzyme is going to catalyze a specific reaction. And the example that we gave before was lactase breaking down lactose. Now, there's three types of specificity that you need to know. And of course, the specificity is controlled by the enzyme substrate complex forming. So you have three types. You have absolute, you have grouped, and you have linkage. What you need to know about absolute is that there is only a single substrate. So an example of this would be urease. It can only break down urea. There's no other a substance or molecule or whatever you want to call it that urease can break down. So that's why we call this absolute, because there's only a single substrate that the enzyme can work on. Now you have grouped on the other hand, which is a group of similar substrates that the enzyme can act on. A good example of this is hexokinase, which we will get into later on, the differences between hexo and glucokinase. And what this does, it's phosphorylating a group of hexose sugars. So it can add the phosphate to to different types of hexose sugars. Right, so this is your group. Then the linkage is there's a specific bond involved. So an example that was mentioned is your chymotrypsin and your peptide bond. Your chymotrypsin over here, the enzyme, is going to hydrolyze this peptide bond. And so we call this a linkage type of specificity because there is a specific bond involved. In this case, the peptide bond. All right. This is honestly really fun. I like this, and it seems very simple and kind of, oh, I'm gonna know this. But usually in the exam, you start forgetting things because of the pressure and all that. So really try to remember this because questions do come from here. Okay, so hold up, there's a question. Isn't chymotrypsin the inactive form though? I honestly don't know. I wouldn't think so because if you remember what we said, it would be the ogen at the end, the zymogen that would be converted into the active form. So I don't think that's the case. But either way, that's not the point of the what we're trying to say here. What we're trying to understand here is that there is a linkage type of specificity where there is the enzyme is specific to a specific type of bond. And in this case, it's just giving you an example. I don't think you need to really know the details of the examples. I hope that's clear. You're welcome. Okay. So six types, major types of enzyme classes. You firstly have hydrolase from the name itself, hydro meaning water. So it is the breakdown of the breakdown when you have addition of water. So you have your reactant plus your water that gives you your product. Your ligase, it comes from the word ligation. So you're adding two. So you have two reactants. You're adding one reactant to another reactant, and you're getting one product. So you have two reactants and one product. Your isomerase, now you can see that it goes from one reactant to one product. So it's a change from one to one. You have one reactant, one product. An isomerase is just simply, it changes the configuration of the molecule itself. So the molecule is the same. It's just a few different bonds here and there that are changed. And so this is what we call an isomerase. It changes from one reactant to one product. Same molecule, just different arrangements. Then we have transferase. Again, the name suggests that there's transferring of something. So there's transferring from this your carbon, your nitrogen, your phosphate, and you have two reactants that are going to turn into two products. And this makes sense. That means one reactant is going to transfer one thing to the other reactant. So now you have two separate products that are formed from your two reactants. So again, your hydrolase is whenever you see water, you know it's hydrolase. Ligase is from two reactants to one product. Isomerase is one reactant, one product. Transferase is two reactants to two products. Now, oxidoreductases. This one, from the name again, is anywhere where we have a redox reaction involved. And so you can see here the NAD plus NADH, the NADH, which are oxidizing reducing agents. So whenever you see those, you know that the enzyme is an oxidoreductase. And then lastly, you have your lyase. Again, this comes from lysis. So you have one reactant that is lysed into two products, or one substrate, one reactant that is broken down into two products. And this is the opposite of the ligase, which makes sense because the ligase you're adding, and in lyase you're splitting. So I hope this is clear. This is quite simple, but this is important to remember because questions again do come from this a lot. Alrighty. Now we have isoenzymes and isoforms. And again, this will be important later on when we talk about clinical enzymology. So let's get this straight. Isoenzymes are different enzymes that catalyze the same reaction that are encoded by a different gene. What does this mean? You have one reaction. Let's say reaction A. It takes you from one reactant to a product. Now, an isoenzyme is it's a different enzyme. So you have subtype A, for example, of the enzyme and subtype B. They're two different enzymes, but they catalyze the same reaction of one product to one reactant to one product. Now, what you need to really, really understand here is that the isoenzymes are encoded by different genes. Now, this is on the contrary to your isoforms, which are the same thing, like essentially the same thing. It's molecules performing the same function, whether it's an enzyme or whatnot. But the difference here is that it's encoded by the same gene. So you need to be able to associate isoenzymes with by being encoded by different genes, and your isoforms being encoded by the same gene. All right. So now for the major part of the first lecture, which is the introduction to enzymes, you have enzyme activity. So there's a few factors that actually affect the activity of an enzyme. The three mentioned over here are your environmental, your cofactors and coenzymes, and then lastly, your enzyme inhibitors. We'll first talk about these two, the environmental and the cofactors and coenzymes, and then we'll get into the kinetics before we look at the enzyme inhibitors. And by the way, the enzyme inhibitors are very, very high yield as well. So you really, really need to know them. Okay, let's start. So for your environmental, you have two environmental conditions. You have your temperature and pH. Let's start with temperature. So we have the graph on the left. So you have your enzyme activity. Again, let's orient ourselves. You have the y-axis, the enzyme activity, and the x-axis, you have the temperature. Now you can see this Q10 over here. This Q10 actually is what we call your temperature coefficient. This temperature coefficient is telling you how much the rate of reaction changes or increases with a change in 10 degrees Celsius in the temperature. So let's look at the numbers now because it's easier to understand that way. You have 30 and you have 40. So that's a change of what? That's a change of 10 degrees Celsius. The change of 10 degrees Celsius, you'll see that there is an increase from 4 to 8 in the rate of the reaction or the enzyme activity, whatever you want to say. It's essentially the same thing. And this difference right here, or this increase, is what we call your Q10. So it is the increase or the change in the rate of the reaction with a 10 degrees Celsius change in the temperature. Normally, your Q10 is around 2 or 3. What this means is that there is a double or triple in the amount of the rate of the reaction, or it's the rate of reaction is doubling with every change in the with every 10-degree change in the temperature. Now, of course, if you look at the graph, you can see if the temperature is too low, the enzyme activity is very low. This makes sense. And then it reaches an optimum. And then after, though, if the temperature is too high, the enzyme activity or the rate of the reaction goes down again. The reason for this is because there is degradation of your enzymes. Okay, and you can see actually the reason that I put 4 and 8 specifically is because of we said that the enzyme coefficient is usually 2 or 3, which means it's a double or triple. So 4 to 8 means there's a double, so it increases by 2. So your temperature coefficient in this case would be 2. Now you have now you have pH. So we talked about temperature, now we have pH. This is just showing you that different enzymes have different optimum pH values. So if you take an example of pepsin, the optimum temperature is around 3. The optimum pH is around 3, and trypsin, the optimum pH is around 9. Now, the differences are, of course, you should already know this from your GI, is because the pepsin, for example, is in the stomach, trypsin is in the intestine. We don't need to get into that, but this is just to show you exactly that there's optimum pH and temperature values for enzymes, and any increase in the pH or temperature is going to degrade the enzyme. And again, you can see that the enzyme activity drops down very low once the temperature is too high. If we take pepsin for an example, once the pH, is too high. Okay. Now, cofactors and coenzymes. When I was studying this one last year, I low-key didn't know what was happening. The names all seem the same: cofactor, coenzyme, co-substrate, apo, holo. So let's get this straight as well. So let's visualize. We have apoenzyme. An apoenzyme is your enzyme alone. It's just the enzyme. There's no substrate, no cofactors, no coenzyme, no nothing. It's just the enzyme by itself. And of course, the enzyme by itself is inactive, right? Now you have your coenzyme over here, this red triangle. Your coenzyme is a non-protein structure, right? That is as compared to your normal enzyme. It's a non-protein structure that is going to help in the binding of the substrate to the uh, active site. So the enzyme substrate complex formation is enhanced. This is what the coenzyme is doing. And you can know this again from the name, because co meaning it's trying to help, you know? Now, when we, and you also have the substrate, the blue ball, we already talked about this. Now, when you have the coenzyme that attaches to the apoenzyme, and by the way, the coenzyme actually attaches to very close to the active site, and you can see this, the active site is right here. It attaches very close to it in order to enhance the binding of the substrate to the active site of the enzyme. Now, this all of together, the whole the enzyme, the apoenzyme, the coenzyme and the substrate all together is what we call holoenzyme. And this, of course, is your active form. And a way you can kind of remember this is holoenzyme is the whole enzyme, right? So that's the active form. All right, let's look at the chat real quick. So isoenzymes share the same chemical rig structure. Isoenzymes can differ in enzymatic or physical properties, but I think they have almost the same structure. Yeah, so let's go back to this real quick. Your isoenzymes are different enzymes. They are structurally similar. That's right. They're structurally similar, and that's why they can actually catalyze the same reaction. Because if it was structurally not similar, that means the active site would be different, and for the same substrate, that active site wouldn't bind. So it makes sense to say that the structure of the enzyme is in fact the same. So isoenzyme, different enzyme, with same structures, catalyzing the same reaction. What you need to know, different gene. Isoform is on the same gene. All right. So we talked about holoenzyme. Now we have cofactors and coenzymes. So what you need to remember from this is if you see a loose bond. So again, you have your apoenzyme over here and your coenzyme. We said coenzyme was a non-protein structure that helps the substrate bind. So that helps the enzyme substrate complex in forming. If we see a loose bond between the enzyme and the coenzyme, we call this a coenzyme. Okay, there's a loose bond. So only one line, you see the bond here. And these coenzymes are the same as the co-substrate. So don't get confused. If you see co-substrate, that's the same thing as the coenzyme, which is the non-protein structure that enhances the ES complex formation. And these coenzymes or co-substrates are usually organic molecules, right? And an example of this is NAD+. Now, on the other hand, you have tight bonds. So there's a strong bond. You see the three lines right in between here. These are bound to what we call prosthetic groups, right? And an example of this would be FAD plus. So again, you might get confused during the exam between NAD, FAD. So remember, loose bond, coenzyme, co-substrate is the same thing. Organic molecule is whatever, I guess. And an example of this would be your NAD plus. Your tight bond, if there's a strong bond between the enzyme, we call between the enzyme and the coenzyme, we call that a prosthetic group, which is this yellow thing right here. And an example of this is FAD plus. Now you also have another cofactor, another coenzyme which we call cofactor. So coenzyme, by the way, is just it's a broad term, right? And then you have the co-substrate, the prosthetic, and the cofactor under them. Your cofactor now is an inorganic molecule compared to your coenzyme or co-substrate, which is organic. And an example of this would be metals. So metals, when they bind to an apoenzyme, this is when we call it a cofactor. All right. All right, now before we get now into the inhibitors, we first need to understand enzyme kinetics, right? So again, let's orient ourselves real quick. We have on the y-axis of this graph, we have the reaction velocity, and on the x-axis, we have the substrate concentration. Now you see that this reaction is without the enzyme, and this is important to understand. You see that there's a linear increase in the reaction velocity and the substrate concentration. What does that mean? That means without the enzyme, by the way. That means that if there is an increase in the substrate concentration, there is a proportional increase in the reaction velocity. So this is why you see a linear increase. Now, if you have a reaction with a fixed amount of enzyme, and it's important that you say that you understand that it's a fixed amount, because if there's a changed amount of enzyme, you won't see this graph as you see here. So if you have the reaction with a fixed amount of enzymes, you now will see you will have the same linear increase for a bit, which is a lot steeper, by the way, than the one without the enzyme. You still have the linear portion, and then it plateaus. It reaches a maximum, right over here. It reaches the maximum, and then it plateaus. Now, this increase is again because of the increase in the substrate concentration, increasing the reaction velocity. But now, because we have an enzyme and it's a fixed amount of enzyme, you reach a Vmax. This Vmax is the maximum velocity, meaning what? It means that this is the maximum. This is the maximum rate of reaction that the reaction can reach in a specific amount of time, right? And now, if you notice, you see that it plateaus. Why does it plateau, you may ask? It's because of this fixed amount of enzymes that we talked about. What does that mean? That means now that because we have a fixed amount of enzymes, all of their active sites are bound to now by the substrate, right? So now you have saturated all of the active sites of the enzymes, and that's why you have that plateau over here after the peak, after the Vmax. And you can also see again over here, which was briefly mentioned by the doctor, your V0, which is your initial velocity. And this initial velocity, the reason that it's at the linear part of the graph and not anywhere else is because that's where you can have a quantitative measure of the rate of the reaction. So this is just your initial rate, initial velocity. All right. Now again, we said your V0 was your initial velocity. And your Vmax is your maximum velocity, which is the rate of the maximum rate of the reaction after a specific amount of time. And then you have your Km. Now this is really important, the Km. When you have a Vmax of here, let's say this is 10. Now your Vmax divided by 2, that means half of your Vmax is what we call your Km. So that would mean if this was 10, your Vmax is 10, that means your Vmax over 2 is 5. So if you go to 5, then you plot it over here and you look at the graph, the x-axis, the substrate concentration at half the Vmax is what gives you your Km. Now, what is this Km? This Km is going to tell you about the affinity of the enzyme to the substrate, right? So half of the Vmax, or the substrate concentration at half the Vmax is what we call your Km. It's the Michaelis-Menten constant or something like that. Just remember as the Km. Now, what you need to know is that a low Km is equal to a high infinity, and the opposite is true. A high Km is low infinity. Let's try to understand this. If we're saying that the Km is the amount, is the substrate concentration at half of Vmax. If we have a low Km, that means that there is less substrate needed to reach half of the Vmax, right? What does that mean? That means that the binding of the enzyme to the substrate is a lot better, a lot stronger, a lot faster. So the affinity is higher because you need less of the substrate of the concentration of the substrate in order to reach the same Vmax, right? So again, if you didn't understand what I'm saying, it's completely fine. Just remember that a low Km means high affinity and a high Km means low affinity. They're inversely proportional to each other. All right. Now you also have a formula here to remember, and this is the only formula you need to remember. There's a bunch of them that are mentioned in the actual lecture, but Dr. Ahmed Al-Jada said that you only need to remember this one, which is the 1 over the initial velocity, the V0, is equal to your Km, which tells you about the affinity, over the Vmax multiplied by 1 over the concentration of the substrate plus 1 over Vmax. Now, why is it all 1 over 1 over? It's because this graph actually, or this formula actually represents the graph of a linear equation. Now you can see here, this graph over here, the enzyme kinetic, this graph, the Michaels-Menten or whatever it's called graph, is not linear. And we said again, it's linear at the beginning and then it plateaus. Now, if you want to convert this graph into this graph, it's the same exact thing. It's just you take the double reciprocal is what this is called, this graph. In order to make the non-linear graph into a linear graph, this is the formula you use. Now, if you notice, if you were to look at it, you can actually notice that this graph follows the equation of a linear follows the formula of a linear equation, which is what from our high school math we remember: y is equal to mx plus c. Y meaning your y-axis being 1 over the initial velocity. So you can see that is in fact true, 1 over v. Then you have your km over your kmax is your m, which is the slope. So the slope you can see over here is the km over vmax. And then you have your x-axis, which is 1 over the substrate concentration. Your x-axis makes sense. And your c, which was your y-intercept. So you can see that the y-intercept over here is 1 over vmax. Now, these formulas are used in order to find when you're doing an experiment, you're trying to find the Km or the Vmax of a specific enzyme, and so you use this because it's a lot easier to quantify than in the nonlinear graph, for example. Right? So please remember this equation: 1 over V0 is equal to Km over Vmax into 1 over the substrate concentration plus 1 over Vmax. Okay. Now, this is where we get into the inhibitors. So it was important for you to understand the kinetics in order to get into the inhibitors. Let's just quickly look at the chat. From the lecture, 7 enzymes slide. Isoenzymes differ in amino acid composition and sequence and multimetric quaternary structure mostly, but not always. They have similar conserved structures. Exactly. And that's what we said. So we said that your isoenzymes have the same structure. And again, why did we say that? Because it's catalyzing the same reaction. So in order for the substrate to bind the active site, the structure has to be the same. Because if the structure is different of the enzyme, that means that the active site is different. And so the substrate won't be able to bind. So I hope that's clear. Can you please repeat kinetics? Okay, let's repeat kinetics. No worries. It's actually good that I'm repeating because this is very, very important to understand inhibitors. Okay, let's start from here. We said that there is a reaction. Let's take the graph on the left. Forget the one on the right for now. We have a reaction without the enzyme. We're looking at the reaction velocity plotted against the substrate concentration. We see that without an enzyme, if there is an increase in the substrate concentration, there's an increase in the rate of the reaction or the reaction velocity. And this increase is proportional. That's why the graph is linear. Right? Now, when we add a fixed amount of enzyme, and again, I can't stress enough on this fixed amount, it's very important. There's a fixed amount of enzyme that you add now. You see that there is the same linear rise, right, at the beginning. Then you reach a maximum point, and then it plateaus, right? So you have the increase, maximum, and then plateaus. Why does it plateau? It's because the fixed amount of enzymes, all of the active sites have been bound to the substrate, so they're all saturated. You don't have any more free active sites, so the rate of the reaction will not be able to increase with this fixed amount of enzyme, right? And we said that the maximum velocity over here of the reaction is signified by your Vmax, which is at the very top of the graph. And then the initial velocity, I don't think I need to get into that. Okay, this is the important part. Half of the Vmax. So we said Vmax is the maximum velocity. If you take half of that, so half Vmax, and you look at the concentration of the substrate at half Vmax, this is your Km. What is the Km? It's the substrate concentration, right? If this was a number over here, you'd see 2, for example. It's the substrate concentration at half of the Vmax. Forget that. If you don't understand it, it's fine. What you need to understand is the association between your Km and affinity. You need to know, and you need to really, really need to know this and remember that your Km and affinity are inversely proportional, meaning if you have a low Km, you have a high infinity. And if you have a high Km, you have a low infinity. And again, if you want to remember it this way, just remember it. You don't need to really understand, but if you do want to understand, so you don't need to, constantly try and remember it, is that low Km means what? That means you need less substrate to get to the same amount of Vmax or the half of the Vmax, meaning that the binding of the enzyme or the active site to the substrate is a lot stronger or faster, just a lot better. And that's why a low Km means high infinity. Okay? Low Km, high infinity, high Km, low infinity. And then the formula, we said that the formula is used to convert that nonlinear graph that we have over here into the linear graph, this one. And we said that it follows of course, the equation of a linear, the linear equation, which is y equals mx plus c. And you can translate this into the graph itself. And please remember this equation again. Can't stress on it enough. Okay, I hope that's clear. Is that clear? Understood. Okay, amazing. All right, now we have inhibitors. This is a very, very high yield concept, and you really need to understand this. Inhibitors are of two types. You have irreversible and you have reversible. Irreversible, why is it irreversible? It's because there's a covalent bond. What do we know about covalent bonds? We know that covalent bonds are strong, strong bonds. And so if there is a strong bond or if there's a covalent bond between the inhibitor and the enzyme, that's what we call an irreversible inhibitor. Okay, it's not being able to reverse, meaning that you cannot remove the inhibitor anymore. Is that clear? Examples of these, nerve gas and pesticides. Now, on the other hand, we have reversible, meaning that it's not covalent bonds anymore. It's not strong bonds. You have in the reversible, you have three subtypes. You have your competitive, non-competitive, and uncompetitive. Now, let's look at each of them one by one. Okay, let's start with competitive. Okay, competitive inhibitors. You need to know that competitive inhibitors are structurally similar to the substrate. What does that mean? That means if it's the same structure as the substrate, that means it's going to bind the active site, right? And this is not the same. If we look at the other ones, we'll look at this in a second and we'll compare all of them. So competitive inhibitors, structurally similar, and so they bind the active site of the enzyme. Now, if we look at the graph, the double reciprocal graph or whatever it was called, you can see that this is your normal. The black one is the normal without the inhibitor, and the red one is with the inhibitor. You will see that, and we said, again, what did we say? We said that this one over here is your Vmax, so the y-intercept is your Vmax, and your Km is the x-intercept. And let me just quickly show you. Yeah, over here we said intercept at the x is -1 over Km, so it tells you the Km, and the y-axis intercept tells you the Vmax. Okay, so you can see if you look at the graph, just forget the text for now. If you look at the graph, you can see that in fact the Vmax is the same. It's crossing the same point at the y-axis, but it's ending at a different x-axis. So that means competitive inhibitors, their Vmax are going to stay. The Vmax for the reaction is going to stay unchanged, but the Km is going to increase. Now, if you really want to understand why it's like that in terms of numbers, let's just look at it real quick. Again, you don't need to know this at all. It will help for the exam in case you do get numbers. But if you just remember competitive inhibitors as structurally similar, binding to the active site, and that the Vmax is unchanged and the Km increases, that's more than enough. But let's just do it for the sake of. So you have your Km. What did we say? We said that this is the double reciprocal, the negative reciprocal. So it's -1 over Km over here. So if we take the normal one, let's say this is more to the left, meaning it's a higher number, a bigger number. So it would be -10, for example. If you have -10, that means is the x-intercept. That means -1 over your Km is equal to -10. What does that mean? That means that your Km will end up being 0.1. I don't know if I did my math correct, but hopefully it is. 0.1 is the Km. Now, if you move more to the right, at the bottom I'm talking about, by the way, because this is actually a left shift. If you look at it from the top, it shifts from the unchanged black to the red. This is a left shift, which increases the Km. If you look at it here, now we went from -10 from the black to a -1, for example. What does that mean? That means -1 over Km is equal to -1. What does that mean? That means that Km is 1. So you change from a Km of 0.1 to a Km of 1. So with the inhibitor, your Km is increasing, right? And this is because this is all math. This is all math. It's because there's a negative reciprocal of your Km. This is why your Km increases when you go more to the right side towards the bottom. But this is a left shift, okay? This is a left shift. So again, I was talking a lot. What you need to remember, competitive inhibitors, structurally similar, so they bind to the active site. The Vmax is unchanged, and you can see this, the graph supports that the Vmax is unchanged and the Km increases. An example, I don't think you need to really remember the examples because it's just thrown in there just for you to understand, really. But you will anyways take all of this in details, the statins and whatnot in your cholesterol metabolism. But anyways, let's just quickly say it. Statins affecting HMG-CoA reductase. Statins are basically just cholesterol lowering drugs. HMG-CoA reductase is the limiting step in your cholesterol synthesis. So this statin is going to competitively inhibit your HMG-CoA reductase, and therefore it's going to inhibit the formation of cholesterol and so on. But anyways, I don't think this is too important, honestly. All right. So we talked about competitive. Remember, again, I'll just say it one more time. Competitive Vmax does not change, Km increases. Now look at the difference between the graphs between competitive and non-competitive. You'll see that now an unchanged, what did we see? We see that at the y-axis, yeah, you see that there is now a change in the y-axis compared to the competitive one where there was no change. So let's just look at this alone before we switch too much. So you have your without the inhibitor, sorry, without the inhibitor in black, with the inhibitor in red again. You see that the Vmax is different. There's a big difference. There's the difference between the Vmax now. So we say that the Vmax decreases. Again, if you look at the numbers, it seems like the Vmax is higher because the intercept on the y-axis is higher. But again, it's the same reason that we said before, because it's the negative reciprocal. That means if you have a higher number that your Vmax is, or a higher number in the graph is going to translate into a lower Vmax. Again, this is just numbers. You don't need to know that, and I don't want to confuse you, which I think I might have already done. But anyways, and so you have your y-intercept. The y-intercept is changed, but you see now the x-intercept is not changed. It's the same. What does that mean? We said that the x-intercept. The x-intercept tells you what does it tell you about? It tells you about your Km. And so from the graph, we can say that your non-competitive inhibitor, it decreases the Vmax and the Km remains unchanged. Again, you really, really need to remember this. By the way, you need to remember the graphs as well. Remember the graphs because sometimes in the question, it just gives you the graph and it asks you what type of inhibitor this is. So remember what the inhibitor is, whether it's competitive, non-competitive, or uncompetitive, which we'll look at in a second. Remember the graph and remember the changes in the Vmax and the Km. So non-competitive inhibitor. Now look at the difference here actually. It binds either to the enzyme or the substrate, but it does not bind to the active site like the competitive inhibitor, right? And again, this is a very important distinction that you need to be able to make. And again, Vmax decreases, Km remains unchanged, supported by the graph. And an example of this, again, not too important to remember if you want to, just for the sake of lead and metal poisons are non-competitive inhibitors. All right. Now you have do we ever care about the slope or do we just look at the intercepts? So like I said, the slope is more important when you're doing the actual experiments and if you want to calculate the Vmax, the Km. But for our case in understanding the uncompetitive, the competitive, non-competitive, and the competitive inhibitors, you just need to look at uh, the Km and the Vmax. But by the way, just just since you mentioned it, the slope is actually going to depend on the Km and Vmax because what did we say? We said that the slope is equal to the Km over the Vmax. So depending on whether the Km increases, decreases, remains unchanged, and the Vmax increases, decreases, or remains unchanged, that's going to affect the look of the slope as well. And I'm sure if we were to insert numbers into here, you would see that the slope increases in this one because Vmax is unchanged, Km increases. But this is not, we're not mathematicians here. Right. So I hope that's clear. Let's look at, lastly, did we do non-competitive? Yes, we did. Let's look at the last one of the inhibitors, which is your uncompetitive inhibitors. Now, uncompetitive, it binds to the enzyme substrate complex itself. So what did we say? We said competitive binds the active site. Non-competitive, sorry, binds either the enzyme or the substrate, and the uncompetitive, it binds the enzyme substrate complex itself. And you can see the graph here is you can distinguish it between the other two very, very clearly because there's no intersection at all. And you see, again, black is your without the inhibitor, red is with the inhibitor. Sorry, and you can see that your Vmax decreases. Your Vmax decreases and your Km decreases. So both both your Vmax and Km decrease in uncompetitive inhibitors. So is that clear? Does anybody have any questions on the inhibitors? Because this is very, very high yield. You need to really, really know this. Okay, so competitive. The inhibitor binds to active site exactly, increases Vmax, increases Km, but Vmax unchanged. Let's look at it. I low-key forgot myself. Hold up. Increases Km, Vmax is unchanged exactly, and it binds to the active site. Good. Non-competitive, the inhibitor binds to the enzyme or the substrate exactly. The Vmax decreases and the Km is unchanged exactly. Uncompetitive binds to the enzyme substrate complex itself, yes, and both Vmax and the Km decrease. Perfect, perfect. Absolutely amazing. That's exactly what you need to remember from these inhibitors. All right, so I hope everything is clear. Any other questions or anything you'd like to say? Sweet. Amazing. Okay, let's do a question. If someone would like to open their mic and read, I would appreciate it. If not, that's completely understandable. I read it. Would anyone want to read? Okay, no worries. I'll read it myself. You are studying the effects of a new drug on the activity of your favorite enzyme. The results of your essays indicate that the drug acts as a non-competitive inhibitor. Which of the following changes to the Km and Vmax allowed that determination to be made regarding the drug's action? So it's a lot of words. All you need to look at here is non-competitive inhibitor and what does it do to your Km and Vmax? So who can answer this for me? Is it A, B, C, D, E? Which one is it? D. It is in fact D. Amazing. Again, this is just memorizing. Non-competitive, we said Km is unchanged and Vmax decreases. Now let's look at all of this together because I talked on this a lot and I probably went around here and there in circles. So let's finally just close this topic off with reversible inhibitors. You have competitive, non-competitive, uncompetitive. The binding of the competitive is to the active site because it's structurally similar to the substrate. The binding of your non-competitive is to the enzyme itself or the substrate. In uncompetitive, it's to the enzyme substrate complex itself. Your Vmax, unchanged in competitive, decreases in non-competitive, and it decreases as well in uncompetitive. Your Km, which is going to tell you about the affinity, your Km increases in your competitive, and your non-competitive, it's unchanged and it decreases in your uncompetitive. And then lastly, whether or not they're overcome by substrate concentration, competitive is overcome by substrate if the substrate concentration is high enough, sorry, and the it's not that is not true for the non-competitive and uncompetitive. I don't think I mentioned this by the way, so let me just quickly talk about this. It's just saying that if you if you now add a lot more substrate, so let's go to the competitive one here. If you add a lot more substrate to this, the inhibition will be removed because there's so much substrate now. So does the inhibitor get removed if there is increased substrate? For competitive, yes. For uncompetitive, no. For non-competitive, no. So it only gets overridden, the inhibition only gets overridden or overcome with an increased substrate concentration in the competitive inhibitors. Okay, let's look at the chat real quick. Okay, we're just saying the answers. Amazing. All right. I hope this is clear. This was probably the most important thing in this lecture, the longest part as well. Now we'll be moving into glucokinase versus hexokinase. So what you need to know from this, and I'm sure you will take the details of the glucohexokinase in your later lectures, the insulin one, metabolism, all of those things in the upcoming lectures. Basically, you will take this, but this is mentioned in this lecture as well, so it's important to know. So glucokinase is found in your liver only, sorry, and your hexokinase is found in all your tissues. Your Vmax for glucokinase is high and it's low for the hexokinase. Your Km though for the glucokinase is high, or sorry, it's the same thing. Your Km is high and hexokinase Km is low. What does this mean? I'm saying a bunch of stuff. What does this mean? I need you to take away from this that because the Km for glucokinase is high, that means that the affinity for glucokinase is low. And the opposite is true for hexokinase. Because the Km is low, the affinity is high because of the inverse proportion, the inverse relationship between your Km and affinity. Now, this actually makes sense if you look at the site, if you look at the tissue and the affinity, you can understand it and remember it that way. Because your glucokinase is only in the liver, that means that it's only effective against the substrates, for example, in the liver. So the affinity wouldn't be too much. But the hexokinase, because it is found all in all your whole the rest of the body in all tissues, the Km needs to be a lot lower, or the affinity, let's talk about affinity, needs to be a lot higher because it's in the whole body everywhere else in the tissue, right? So I hope this is clear. This is just the graph to show you the Km and the Vmaxes. So let's look at it real quick. You have your glucokinase. You see that the Km is your Km is right here, or sorry, your Km is over here. Then you have your hexokinase. Sorry. Let's redo that. So let's orient ourselves first. You have the enzyme activity on the y-axis, your glucose concentration on the x-axis. Let's look at the red graph. And this is for myself, by the way, because I was looking at the wrong graph. You have the red graph, glucokinase, your Km is right here. It's 10. And your Vmax is around here, whatever. We're not too concerned about Vmax because Km is the most important factor because that's how you determine the affinity, like we said. Your hexokinase, on the other hand, you see that the Km is right here. This blue graph, the Km is a lot lower. So Km for hexokinase is low because the affinity for hexokinase is high, and the opposite is true for glucokinase. Glucokinase, low affinity, hexokinase, high affinity. So you can remember this, hexokinase, the H in the hexo is for high affinity. We're talking about affinity because that's the most important aspect. Okay, so we have a question. Vmax would be 20 if Km is 10, since Km is half of Vmax. Okay, again, Vmax would be 20 if Km is 10. Exactly. Amazing. You understood it absolutely perfectly. Why is that? Because we said the Km is half of the Vmax, right? Okay, I hope that's clear. Right, there's something else. In MOL 1, doc said hexokinase has low Vmax and low Km. That's what we're saying as well, yeah. The Vmax, I don't know if you're asking this as a question or correcting me. Sorry that I don't understand, but the Vmax for hexokinase and the Km for hexokinase are in fact both low. Okay, so I hope this is clear. Again, very important that you're able to associate your affinity with the Km and how it changes. All right. Now we have pathway regulation. So how are the products regulated in your pathway? Because of course, sometimes you would want more of the product, and sometimes you would want less of the product. So how is this regulated? It's regulated by our feedback loop. So we have two types of feedback, or three actually, two more of the important ones, and then the third one is kind of a constituent of both of them. So you have your negative feedback and you have your positive feedback. Let's look at negative. Negative is where a product towards the end of the reaction, or as you're moving across the reaction, is going to inhibit the reaction beforehand, before that part. Let me say that again. I'm tweaking right now. Sorry. Negative feedback. You have a product towards the end of the reaction is going to inhibit the reaction towards the beginning. So you see the loop right here. You have your product C is going to inhibit the reaction from A to B, right? Which means that now if we inhibit the reaction of A to B, B won't be able to convert to C. So you decrease C that way. And that's why we call it negative feedback, because you're going to decrease the product towards the end. The opposite is true for positive feedback. You have a product towards the end of the reaction that is going to activate, stimulate, or it's going to positively affect the reaction that is prior to that product. And again, you see the same product C is now activating the reaction of A to B. And that means if we can have more, or stimulating, if you have more stimulation of A converted to B, that means you have more C. And this is why we call it positive feedback. Now, a feedback coupled to another reaction is just showing you that you have one reaction going on, which is the A, or the step series of reactions from A all the way to C. And you have another reaction that happens within it, which is your ADP being converted into ATP. And you see this a lot in your glycolysis and all of these things. You see this a lot. And I'm not sure if you should have actually studied this already in MOL 1. So it should be a little easier for you to understand. So you have another reaction, which is the ADP being converted to ATP, which will now either positively or negatively affect the main reaction itself. So in this case, ADP would stimulate, so you would have more. ATP would inhibit, so you would have less. Negative feedback, positive feedback, because there's a secondary reaction happening that is affecting the primary reaction in this case, which is our A to B to C. All right. Let's look at the chat real quick. No, I'm trying to remember. Thank you. Okay, I'm sorry. I understood it in the wrong way. It's great. Yeah, that is right. So high levels of thyroid hormones inhibit hypothalamus, means negative feedback. This is crazy, honestly. This is ERP stuff. I don't remember this myself. High levels of thyroid inhibit the hypothalamus, so this is negative. Exactly. If there's an inhibition, that means there's going to be decrease of the thyroid from the hypothalamus, and so negative feedback. This is too complicated, honestly. I don't remember this myself, but you get the idea, and I'm happy that you understand that. All right. Now you have allostery. I think we're getting towards the end of kinetics, enzyme kinetics, which is the lecture two. So we have allostery. What is allostery? What is an allosteric enzyme? Sorry, it is an enzyme that catalyzes the committed step by binding to other than the active site. So what did we say before? We said before that your cofactor, for example, coenzyme, it binds close to, or it even binds the active site itself in order to allow the initiation or the formation of the enzyme substrate complex. Now, this allosteric enzyme is going to bind to a site other than the active site, so somewhere else. Right? And it catalyzes the committed step. What is this committed step, you may ask? The committed step is the first reaction to a unique pathway. So, if we were to look at the example of this one again, A going to B going to C, I would assume that the committed step over here would be the reaction of A to B. Why? Because this is the first reaction or the first step in order to go to the unique pathway of making C. So this is your committed step. Now, your rate limiting step, don't get confused between these two. The rate limiting step is what determines the reaction, what determines the speed of the reaction. Why? Because it's the slowest reaction. So understand the difference. Committed step is the first reaction or the first step that takes you to a unique pathway. Your rate limiting step is the slowest reaction in that pathway. All right. Now, if you look at the graph again, this is really important. Is again, let's orient ourselves real quick. We have the reaction velocity on the y-axis, and the substrate concentration on the x-axis. It's the same graph that we had, or the same plot that we had before. Now you realize that the graph over here, the plot over here, is not the same as we saw before. What is the shape? This is what we call a sigmoid shape. You see, it looks like an S, a sigmoid shape. Now this is in comparison to for example, this one. If we look at it, the hexokinase. Don't look at glucokinase because it low-key looks like a sigmoid. Hexokinase, where it increases and then plateaus. It's not the same. Why is this? This is because of something that we call cooperative binding. Now, the best example I can give you of this, of how to understand this, is by giving you the example of hemoglobin. Hemoglobin, we know it's made up of many different subunits, 4 subunits, I think, 2 alpha, 2 beta. Not important. It's made up subunits. That's what you need to know. These subunits are now going to bind substrates differently, and this is what we call cooperative binding. There's subunits in the end of the enzyme itself that binds to the substrate, and this is why this graph looks the way it does. This is why it's a sigmoid graph, because of the cooperative binding or cooperative binding. Now, another thing that you need to know, and again, you can remember this with hemoglobin as well. I'm not sure if you've taken this lecture already before, or an FND or whatnot, but anyways, that's not important. You have 2 states of enzymes. You have your T state, which is your tense, T for tense, and you have your R state, which is relaxed. Now, what you need to remember, again, this is all being associated with affinity, because it's so important to understand whether the affinity of an enzyme is high or low. Km we talked about, we're looking at affinity. Again, now we're looking at the tense or relaxed, we're looking at the affinity. You need to remember that your T state, or the tense state, is going to have low affinity. Why you might ask yourself, why is that the case? I want you to imagine it this way. Let's say I am the enzyme and I'm tense, right? I'm tense. I don't want anything to bind to me, and that's why I have a low affinity. I don't want to form enzyme substrate complexes. I don't want to bind a substrate or a reactant. So in tense, low affinity. The opposite is true for your relaxed state. You're relaxed. I'm here. I'm the enzyme. I'm relaxed. I'm like, all the substrate come to me. I'm relaxed. I don't mind. This is why the affinity for a relaxed state, or the R state, is high. I hope this is clear. Now, the inhibitors, now listen to this, this is important again. Inhibitors stabilize the T state. Again, think about it. Inhibitors stabilize the T state. What is the whole point of inhibitors? It wants to, of course, inhibit, who would have thought, the enzyme substrate formation, complex formation. Why? So that the reaction doesn't happen. So if we want an inhibitor, what does the inhibitor do? It's going to want to decrease the affinity, so less of the substrate binds. And this is why it stabilizes the T state, because the T state is low affinity, right? So inhibitors are going to stabilize, or it's going to favor the T state, so that the affinity is lower, and therefore the reaction cannot progress, because there's not enough reactants binding to the active site of the enzyme. Sorry. Now the opposite is true For activators, what do? They want the reaction to progress. They want it to go on, and therefore they're going to stabilize the R state. What is the R state? High affinity. So if I want more of the R state, favor the R state, more affinity, the reaction is going to be able to progress a lot faster. So, is that clear? Inhibitor decreases affinity. It favors T state. Opposite is true. So inhibitor, it decreases the affinity. Sorry, exactly, and it favors the T state, which is the tense. I'm tense. I don't want any substrate to bind to me, and the opposite is true. Amazing. You're doing great, guys. You're doing great. All right. So the reason this doesn't have a title is because this is low-key just miscellaneous information. I don't know whether this is really important for you to know. It is mentioned in the slides, and the doctor doesn't, talk about the slide quite a bit for quite a long time. So I just thought I would mention it. I don't personally think it's important. Do with that what you will. But let's just look at it. Measuring enzyme activity. How is enzyme activity measured? So it's done by something that we call photocytometry or something like that. Basically, you have a substrate that has it's called a chromogen. A chromo meaning color, uh, and so you have this chromogen. When it binds, it's going to emit the color. You're going to see the color based on frequencies. Okay, and so there's two types of measurements. You have discontinuous and you have continuous. Discontinuous, you have the substrate and the enzyme mixed. So let's say I have a container, I add the enzyme, I add the substrate, and I mix them. And now I set a time for 10 minutes, for example. And after 10 minutes, I look at what the activity of the enzyme was, or what the reaction of the enzyme was, or how much light was emitted because of the chromogen. Now, for continuous, you have a substrate and the enzymes are mixed. So you have the same container. I add the enzyme, I add the substrate, I mix them. But now, instead of putting a timer for 10 minutes and then marking down the activity after 10 minutes, I'm going to put a timer for 1 minute. I write down after 1 minute. I do at the second minute. I write it down. And so you're continuously measuring the product, or you're continuously measuring the enzyme activity, which is, by the way, measured by the product because we said chromogen, yada, yada, in intervals, which is the important distinction here. So discontinuous, it's okay, no. Discontinuous, you have after a set amount of time, continuous in intervals. And then I also noticed that the doctor mentioned a few clinicals. There was two clinical slides. One was about gout, where he talked about if there's a problem in the allosteric enzymes inhibition, then it leads to gout. And there was another one about ALD, uh, ALDH, and alcohol and all of that. I don't think you need to know this for now because you will have separate lectures talking about gout specifically in nucleotide metabolism towards the end of the semester. And you will have a separate lecture again towards the end of the semester, alcohol metabolism, where you will talk about the details of all of this. And this is where the infamous story of, Japanese or Asians not being able to tolerate a lot of alcohol will come. So do look forward to that. But anyways, I didn't add this because I don't think it's really important for this lecture. If you want to go through it, it's just two slides in the doctor's lectures. Feel free to do so in your own time. And I'm sorry I didn't add them on the slides here. All right, checkpoint. Finally, we're done with enzymes 1 and 2. We're done with intro to enzymes and enzyme kinetics. We only have clinical enzymology left. Is there anything that you guys are confused with, that you want to ask, that you want to confirm at all? We took in fructose metabolism, high uric acid causes gout. Exactly. Gout is because of high uric acid. And the problem in that reaction pathway is that there is no allosteric enzyme. And so there is an increased amount of the uric acid in the pathway, or an allosteric inhibitor, sorry. So any questions at all from the previous slides that we've had? And we're doing pretty great on time, 1 hour, 3 minutes. We only have a few more slides left about clinical enzymology. Good so far? Amazing. All right. All right, sorry about that, guys. Let's move on. Clinical enzymology. Now, honestly, this lecture is very fun, to say the least. But Dr. Raja in her lecture has a lot of information that is really not needed at all. You don't need to know half of the stuff that is there. There's maybe 3 or 4 slides that are really, really important for you to remember, and that's the biomarkers and what enzyme is associated with what disease. So that's what I focused on here personally. Again, I'm really sorry that I didn't include all the other stuff. I just don't think it's important, and it's not something you're very likely to get asked on. If you look at the past questions, um, you will see that most of the questions, or all of the questions actually, are from the biomarkers. So myocardial infarction, what enzyme, hepatitis, what enzyme, and so on. So I do apologize that I didn't include those. Anyways, let's start with clinical enzymology so we can finish within the next 15 minutes. We have your first, so the first two slides are your enzymes and what is associated with, what disease is associated with the enzyme. So let's start. ALT, it's specific for liver diseases. Okay, it's specific for liver diseases. Now, you'll see now, if you look at AST now, it's in M, you can have it in MI, which is myocardial infarction, and you can have it in liver diseases. So that means AST now is not going to be specific for liver diseases. Why is that? Because you find it in you can find the enzymes will be found in there's more different enzymes that are found, sorry, in the same disease. You will find multiple enzymes. The only enzyme that is specific, that you will only find in the liver and not MI, for example, is ALT. So ALT is specific to liver enzymes. AST, you have myocardial infarction and liver diseases. CK, you remember before when we were talking about isoenzymes, I was saying that we're going to get to that. This is exactly the point I was trying to get to. CK and its isoenzymes. So you have three different subtypes or three different isoenzymes of CK, which is creatine kinase. What did we say? Can someone remind me what we said isoenzymes were? Who can tell me what isoenzymes were? We said isoenzymes are structurally similar and they catalyze the same reaction, right? Isoenzymes are enzymes that share similar structures and they're made by different genes and catalyze the same reaction. So look at this. Thank you, by the way. If you look at the isoenzymes of CK, you have three isoenzymes. You have CK1, you have CK2, and CK3. CK1 is two subunits. It's BB, a subunit of B and another subunit of B. CK2 is a subunit of M and a subunit of B, so MB. So CKMB is the same as CK2, CKBB is the same as CK1, and CK3 is the same thing as CKMM, two subunits of M. Don't ask me what M and B are because I honestly do not know. Okay, but anyways, so what you need to be able to associate with CK1 is CNS diseases. And this means that what does CNS disease means? It means it's a disease in the brain or the spinal cord, for example. Let's stick with the brain. So that means CK1 is found in the brain. So if you see elevated or abnormal levels of CK1, that takes you towards a diagnosis of a CNS disease. Now for CK2, which is your CKMB, remember CK2 is CKMB actually. This is a biomarker for myocardial infarction. And then CK3 is for muscular disorders, many different muscular disorders. Now, LDH, lactate dehydrogenase, I think it's called. It is for LB, their biomarkers, sorry, for MI and muscular dystrophies. So you can actually see that there's a lot of markers that are for more than one disease. So you can see MI has AST, CKMB, LDH. You see liver diseases in ALT, AST, ALP. And this is just when you go into the clinic, actually, you're going to now not base your diagnosis off of one biomarker. No, you're going to order multiple biomarkers. You're going to order this, this, this. You're going to rule out anything that you see according to the biomarkers, and you're going to give the diagnosis based on these biomarkers. So you don't make a diagnosis based off of just one single biomarker. No, but rather you need multiple biomarkers in order to give your final verdict of what the disease is. Right. So we talked about LDH being myocardial infarction and muscular dystrophies, and we'll get into the details of LDH as well, the isoenzymes. And then you have ALP, which is for obstructive jaundice, bone diseases, and liver diseases. So again, you see this a lot. There's a lot of enzymes where there is liver diseases, but specific ALT. Okay, and we have five more. So that was the first five. Now the second five. Acid phosphatase. This is for prostate cancer because you see this enzyme in the prostate. Sorry. So that means an abnormal level of acid phosphatase will indicate a problem with the prostate. So prostate cancer. Amylase and lipase. These are important. You see, amylase and lipase, they actually both are found in pancreatitis. But the difference is actually that in amylase, it also it's also seen in salivatory diseases. Sorry, I don't know how to pronounce that. It's also seen in salivatory diseases, whatever. And in lipase, it's only for pancreatitis. Now you have this gamma something transferase, the GGT, and you'll find that in obstructive jaundice, again, liver disease, and chronic alcoholism, right? And you can see how these kind of go together. They're all like the hepatobiliary tract. And so it kind of makes sense that they all are associated with GGT. And if you see ALP as well, obstructive jaundice, liver disease, so you can kind of catch on that way. All right, and lastly, you have the ceruloplasmin, which is for Wilson's disease. I think it says a deficiency in copper, zinc, something like that. I'm not too sure, honestly. But just remember ceruloplasmin and Wilson's disease. All right, is that clear? There's honestly nothing much I can do other than read off the slide for this because it's just memorization. You just need to know what biomarker is associated with what disease. All right, so before we were looking at the enzyme and what disease it's found in. Now we're going to look at, to kind of put everything together, what the diseases and what enzyme you'll find. So MI, LDH1. What did we say about LDH? We said LDH will find in the MI and muscular dystrophies, specifically LDH1, which is an isoenzyme of LDH. So LDH1 is specific for MI. And remember that one is very, very important. And CKMB, we talked about this as well, which is the CK2 in myocardial infarctions. And this is for repeated infarctions. Now, let me get to myoglobin first, and then we'll see. Myoglobin, again, this is released for MI and for early diagnosis. Now, you actually see in brackets, I wrote these specific, and okay, sorry, we have troponin, we have one more, troponin 1 for late diagnosis. Now, you can see, okay, this one is specific LDH, CKMB is for repeated infarctions, myoglobin for early diagnosis, and troponin for late diagnosis. You might ask yourself, all these cardiac enzymes or all these enzymes that are being released in a myocardial infarction, why is this one specific? Why is this one more important than repeated infarctions in early and late? This is because of the level or the time that these increase. So you will see, for example, that myoglobin is increased in the first, for example, 6 to 12 hours. I'm making these numbers up. I'm not 100% sure if that's true, but it's increased basically very early on. So that's how you know this is an acute myocardial infarction if the myoglobin is high. The opposite is true for troponin. Troponin, there's a late diagnosis. This is for late diagnosis of myocardial infarction. What does that mean? That means after 48 hours, let's say 48 to 72 hours, that's when you will see the troponin increase, and that's how you can say that this is not an acute myocardial infarction, right? And then the last two we have is liver diseases. ALT is specific. ALT is specific for the liver. You have AST, ALP, GGT, LDH5. Now you need to remember all of these AST, ALP, and GGT. But now the distinguishing factor between LDH and liver disease and LDH and myocardial infarction is again the isoenzyme, and this is why it's so important to know. Anyways, LDH5, liver disease, LDH1, myocardial infarction. How can you remember that? The LDH1 looks like the I in the MI. So myocardial infarction, LDH1, liver disease, LDH5. Please remember that. And then lastly, bone diseases. We already talked about that is ALP. Yeah, ALP, we said bone diseases is one of them. All right, that's it for the enzymology pal. I hope you guys found this beneficial. Does anybody have any questions before we start? Okay, so we have someone summarizing it. Muhammad, I really like this. CK1, brain, exactly. CK2 or CKMB is the heart, perfect. They are isoenzymes, amazing. CK3 for the muscles, yes. LDH is the MI, but remember, LDH1 is for MI, and LDH5 is for liver diseases. Remember that. Amazing. And ALT is for obstructive jaundice. Obstructive jaundice, GIT disorders, exactly. So the hepatobiliary tract problems, bones, and liver. So I don't think bones. Yeah, bones is ALP, not ALT. Yeah. Uh, I'm not sure if you can put bones in another category as well, but what I have written here, I'm going based off of that. ALP is for bones, so please don't get confused with that. ALT, let's do it again, specific for liver diseases. Right? Liver diseases, ALT. Then you have GIT bones and ceruloplasmin, or whatever this was called, ceruloplasmin for Wilson disease. Amazing. Okay, so that's it. If you have any questions, you can go ahead, you can ask in the chat if you have any questions at all. If not, then I really, really appreciate your time, guys, today. It means the world. Please scan this QR code. I think you're going to get a few questions. Do solve them. Please solve as many questions as you can for MOL 2, right? The infamous statement of Dr. Ahmed Al-Jada, where he says 60% of you are going to fail MOL 1. Please don't let that happen for MOL 2. Inshallah, I wish you guys the best of luck. Of course, if you have any questions at all about this lecture, about MOL in general, about anything medicine, non-medicine related life, I'm here for you. My number is on the first slide. Feel free to text me at any time, call me, whatever you want, whatever makes you comfortable. Um, and I think I'll end it with that. Thank you so much, guys. Assalamu alaikum wa rahmatullah.